So we've been talking about chaos, and I want to complete the discussion with a real-world example, a physiological example, which is cardiac arrhythmias. But first, I want to remind ourselves of what we saw as the mechanism of chaos in the dripping faucet and then in the kicked spring system. And that is this, that when we have a process that is being stimulated nice and slow, and these are the stimulus events, and there's a nice time interval between them, and you had an action in response to the stimulus, and you had a recovery in response to the action. And then everything waited. And then you had another stimulus and another response, and everything waited. That in that circumstance, everything came out periodic. But when you just increased the driving frequency, as they say, then now you had a situation where the next beat is encroaching. I said beat, giving it away. The next action is encroaching on the recovery from the previous. And that is going to create a mechanism for irregular action or chaos. So I want to talk about the heart, and we have to review some very basic facts about the heart. If this is your heart, and there's the left ventricle and the right ventricle, and these are, of course, cavities in the ventricular muscle, the way that the heart beats is that each beat consists of a wave of contraction. The muscle cells here are all contracting, and that creates a wave of contraction passing up the heart, and that wave of contraction then ejects the blood to the body or to the lungs and creates what we call the heartbeat. Now, next question, what creates that wave of muscle contractions is another wave. It's a wave of electrical activations by the cardiac muscle cells, which are also electrical, as indeed all muscle cells are electrical. And in every case, the electrical activation creates the contraction. Contraction is a response to the electrical activation of the cell. So we're going to have a wave of electrical activations passing up the heart and creating the wave of contraction. And then we have to ask, well, what creates the wave of electrical activation? And in order to understand that, we have to understand some basic facts about the cardiac cell. So the cardiac cell, like the nerve cell, like many other cells, is a highly polarized and electrically charged object. Typically, outside in the environment, sodium ion is very high. Think of the seawater, which is salt. And potassium ion is in very short supply in the general environment. Inside the cell, it's the other way around. Sodium is at very low concentrations, and potassium is at very high concentrations. And that imbalance is maintained by pumps. And those pumps are constantly working to shovel sodium out of the cell and to shovel and sequester potassium inside the cell. And these pumps require energy. In fact, that's where your lunch is going. Your lunch is going to power the pumps that are maintaining these ion imbalances inside the cell. Then, what happens when the cell receives a little bit of stimulus is sodium starts flowing into the cell, and as the sodium flows into the cell, the voltage changes because sodium is an ion. The flow of ions is a current. The flow of charged particles is a current, and that current changes the voltage of the cell. And if we just put V over there, when a stimulus is achieved by the cell, the inrush of sodium creates a rise in voltage in the cell. Then there's an outflux of potassium from the cell, and that brings it down again. But now we have sodium inside the cell, which we don't want. We have potassium outside the cell, which we don't want. And now the pumps have to go to work to pull all that sodium out and to pull all that potassium in. So the sequence of activation of the cardiac cell, or of the nerve cell for that matter, is sodium rushes in and brings it up, potassium rushes it out and brings it down, and then pumps go to work restoring the imbalance. That's the single cell. Now, the wave is created when one cell communicates its voltage change to the neighboring cell, which then is stimulated, and like a string of dominoes, a wave of electrical activation is created subsequent to a single stimulus that will spread through the heart. So now we're going to do an experiment. We took a chunk of animal heart, put it in a dish, put electrodes in it so that we could record from it. And we also used a drug. We gave a drug to the tissue. The name of the drug is Wabane. It doesn't matter. But I'm going to tell you what it does. If you remember sodium rushes in and then potassium rushes out, and then the pumps go to work. This particular drug poisons these pumps and retards their effectiveness, makes them less effective. The first consequence of the drug, when it's administered in fairly low doses, is that the pump is sort of blocked in a slightly open state. And so sodium keeps leaking into the cell, thereby creating another electrical activation wave automatically. That's not a normal process. But what you see is that under these low doses of this poisoning drug, the cells suddenly become spontaneously active. And you see the string of action potentials that are generated. Action potential is the fancy word for the voltage depolarization, the voltage deflection. You see this string of electrical activations which are happening autonomously in the heart as sodium leaks in, sodium rushes in, then potassium rushes out, then the pumps go to work, then sodium leaks in again. So the result is this periodic looking train of electrical activations. Now we're going to verify that that's periodic because we're going to make that Poincare plot that we were talking about. We're going to take as our data the activation time, the interactivation times. So this is going to be I1. This is going to be I2, I3, etc. And it's probably sounding familiar because we developed the same kind of plot for the interdrip intervals of the dripping faucet. And we're going to do it now again for the interbeat intervals for the cardiac preparation. And then we're going to do a Poincare plot. And the Poincare plot is going to be In plus 1 as a function of In. And the question is, what is it going to look like? Now, in a periodic, let's again always draw that 1-1 line. In a periodic activation, like you see here, I1 equals I2 equals I3 equals In for all n, and therefore the Poincare plot, remember what these are called, they're called Poincare plots. The Poincare plot of the periodic activation is just one big dot sitting right there on that line because In equals In plus 1. Then we started to increase the amount of drug. And as those pumps got more poisoned, we began to see a very interesting phenomenon, which you see here. And it is clearly a period 2 rhythm. You see a short interbeat interval followed by a long one, followed by a short one, followed by a long one. And if we look at that time series, we see here I1 is short, I2 is long, I3 is short, I4 is long. And if we make the Poincare plot of that, In plus 1 against In, you see the result. And it is basically a short, here is a long beat followed by a short beat. And then the next point in the Poincare plot is a short beat followed by a long beat. And then a long beat followed by a short. And the plot will alternate back and forth between these two dots. And that is a period 2 rhythm. Now, if we give more drug, we see this. The period 2 rhythm has doubled in period again. It is now a period 4 rhythm. And you see 1, 2, 3, 4, 1, 2, 3, 4. There's a more complex oscillation. And when we make the Poincare plot, you exactly see 4 dots. And we will visit those 4 dots in succession with each of the 4 beats. And that is a period for rhythm with more drug. So then the next thing is to give even more drug. And when we give even more drug, now we see an irregular rhythm. We see intervals of all sizes following each other. I mean, stare at that. What do you want to say? It looks pretty random. They're very irregularly following each other. But when you make the Poincare plot of next interval against previous interval, look at what you see. So that's not the mathematical model of the parabola, but there is a parabola there. It's very clear. That is not a random scatter of data points. It has a very strong suggestion of the parabola. You will also notice that at the place where the data curve intersects the 1-1 line, those are the equilibrium points of this system if it were a dynamical system. You'll notice that the data curve, that its slope at the equilibrium point is steeper than 1, which is a necessary criterion for that equilibrium point to be unstable. And indeed, that equilibrium point is unstable. And we have therefore produced evidence that this irregular beating is in fact chaos.